Hello, everyone, and welcome to this webcast regarding advantages of digital PCR and its application, the power of partitioning. My name is Heinz Löfgren, and I'm the Marketing and the Development Manager for digital PCR in EMEA in Europe for QIAGEN. My background is I have 6 years of experience with digital PCR, and I have been working with QIAGEN since 2 years back. Just to start this introduction, first of all, the system I'm going to present to you and also some basic of the techniques is intended for research use only. If we look for the agenda, we will start with a slight introduction of the basics and principles of the digital PCR. After that, we will follow with a change and let's say a difference between the different PCR methods. We will continue on with the digital PCR system technology and specifications from QIAGEN. We will continue with the power of partitioning and the enrichment effect, and this is actually everything behind the digital PCR. We will continue with applications, tips and tricks, and the hallmarks of PCR, and then after that question. So let's start. So first of all, when it comes to QIAGEN, QIAGEN is a company that has a great expertise in sample preparation and also sequencing, library prep, and PCR. And now, since 1 year back, we acquired the digital PCR technology from Formulatrix and the Constellation system that we now have renamed to QIAGEN. So, what is digital PCR? It's for some considered as the third generation of PCRs. So what does that mean? Well, if we go back to 1983 when Mullis actually invented PCR, what we did in 1983 was that we run our product, we created primers and an amplicon. We did the PCR, we ran it in a gel, and we stained it with a DNA binding dye like ethidium bromide, and we take a look at it, and hopefully we had our product. That is called endpoint PCR, and this is very important that you know that this is an endpoint PCR. It just gives you a yes or no answer. So in basic principles, this is our digital PCR, but in a very bad resolution. The second generation of PCR was when we did advance the PCR into real-time PCR. And then we started to use intercollecting dyes or hybridization probes or hydrolysis probes or molecular beacons or scorpion probes to actually see what happens after each cycle. This is very important because real-time PCR gives us relative quantification. This means that we actually need something to compare against. We also use standard curves. We rely on efficiency because we need to know that every cycle has been amplified or duplicated perfectly to 100%. Many people now, when we switch to digital PCR is still, and this is very important, an endpoint measurement. We are using the chemistry for qPCR, and this is why many people are doing a very common mistake, thinking that digital PCR is based on efficiency, which is not. It's an endpoint measurement. But because we are using the intercollecting dyes or the chemistry for real-time PCR, we can actually do the endpoint measurement, or what we did in a gel back in 1983, now in 2020, in a much higher resolution. But it's an endpoint measurement. It means it's not based on efficiency. And of course, since it's a yes or no answer, it's either positive or it's negative. It's nothing in between. And it gives us absolute quantification. It follows also the normal PCR setup. So we actually have a bulk sample but we actually have our sample, our template, where we actually put in a master mix and our primers, which are specific for the sequence we are looking for. The only difference, we are not performing the PCR in a bulk. We actually do that in partitions. So we actually partitioning the sample to actually get an enrichment factor, and this is what this presentation is all about. I will come back to that later. But after the partitioning is done, then you do an endpoint PCR, and then we'll take a look at the outcome and the result, which is absolute quantification. So when we take a look at the dPCR principles, every digital PCR platform is using Poisson statistics and the law of random distribution. And we are doing it statistically also by measuring this in a 95% confidence interval. So what does this mean? It means that for a number certain of molecules that are randomly distributed into these partitions, we have some space that we can work within. But of course, it also sets some limitations. Some of these limitations is actually that we have, let's say, a Goldilocks zone even for digital PCR. If we refer to the Goldilocks zone where we are in a qPCR, that means that we want to aim to be, let's say, not in the upper end of the standard curve in the qPCR and not in the lower end. We want to be somewhere in the middle, so we know that our assay actually works. This is a little bit the same thing. We want to not have too few molecules, because then we perhaps cannot trust if they are false positive or not. This can be checked, of course, but we also don't want to get to the other side where we have an oversaturation, because since it's Poisson statistics, we also go by this very simple formula where we have target copies per partition, which is the minus logarithmic of 1 minus p. And basically that means the fraction of negatives and positives. So we actually need to have both positive and negatives to get the concentration, and this is very important. So the systems normally, and what we have at QIAGEN, is about 5 orders of magnitude. That means from one positive molecule to, let's say, 150,000 something amplified targets. So if we look at the different PCR methods, so what is the major difference? So both PCR and qPCR is run in bulk. And as I already mentioned, in digital PCR, we do partitioning. And here is where companies do it differently. So there are different number of partitions in different vendors' machines. And I will explain what that means to you as well when it comes to the number of partitions. We will have 2 sets of plates. We are doing our partitioning on plates, which one is actually 8,500 partitions and the other one is 26,000 partitions. When we look at the offering, the QIAcuity systems from QIAGEN, here you see that we have actually 3 formats. We have 2 smaller systems, which actually have 5 target multiplexing and 2 target multiplexing. The other 2 has a 4-plate system where it can run 4 plates simultaneously, and the other one is actually an 8-plate system which can run 8 plates simultaneously. Both our plates fits on all systems. The only difference in between them is the throughput that we have and nothing else. And we run our first plate within 2 hours, and we can actually run up to all 4 plates within less than 6 hours. And in this one, which has 2 PCR machines inside, can actually run all 8 within 6 hours. So if we look at how our plates are built, so we are doing our partitioning inside our wells. So we have an input well, which is actually connected to microfluidics, and in these microfluidics, we have some nano compartments. This is a grid which actually are connected to each other at this stage. They also end up in an air vent because we need that to be able to prime the well with our sample. So if we go into what the benefits of partitioning is actually is. So if we look at a real-time PCR, when we have something that are exponential, so basically the light emitted is actually based on the concentration in a normal real-time PCR. So the more concentration we have, the more light we have, and also the stronger these CT values or CQ values is, they're coming up very early in a qPCR if you have a lot of product. When we do this partitioning, so the partitioning is different. Here we either have a signal or we don't have a signal. So it's because we either have an amplification of our target or we don't. So this random distribution is actually making this enrichment effect taking place. But mark my words, just because we have a higher precision to amplify our target doesn't necessarily mean that this is more sensitive than a qPCR or a standard PCR, because these are still a PCR technology, which means we are bound to the same principles. It means we can amplify one target molecule, but so can a qPCR, so can endpoint PCR. The problem, or let's say what the benefit is with this partitioning, is that by partitioning, we increase the chance of finding this molecule, which is rare. And also, therefore, we are actually in a very good way have more precision, and this is very crucial. So the benefits of partitioning. So in this example that we have here, we have two conditions. The first one is a 16 microliter reaction, a bulk reaction. And in this 16 microliter sample, we have two molecules inside. So the effective concentration in the bulk sample is now we have one molecule in 8 microliter of sample. So if we take this pure sample with only those molecules and partitioning it in 1 microliter partition in this particular sample or example, you will see that these two molecules randomly distributes out to these partitions. And of course, since we have two, they will end up in each partition, so they will not end up in the same partition. That could potentially happen, but this is very unlikely due to the statistics behind it. So now we have an effective concentration of one molecule in 1 microliter. So that means we effectively increase the concentration of this molecule in this particular case by 8 times. So if we take this same setup, I mean, in this pure setup, these two molecules will probably be found by real-time PCR and will come up at, let's say, CQ of 38 or 37 or something, because they have no competition. It's very pure, but they will likely be found. But in the next example here, when we are to the right, when we have these two molecules surrounded by competitive molecule, this can be two mutation, it can be virus, it can be pathogens, it can be anything that we are looking for. But this surrounding molecules or inhibitors will prevent us to find these two molecules inside this tube with a real-time PCR. But since we're doing this partitioning, all these molecules, competitive molecules, inhibitors, and anything that can interrupt us to amplify our target will randomly distribute. So let's say we have two molecules here, or let's say we have 1 in 1,000 or 1 in 2,000 of a mutant compared to wild type. In this bulk sample, you will never find it. But since we do this partitioning now, instead of being 1 in 1,000, these molecules can be 1 in 2, or by itself, or 1 in 3, 1 in 4, or 1 in 5. And we will always amplify this molecule now because there is less competition, less inhibition inside these partitions. So this just means that we have increased our chance of getting the molecule amplified. The only problem we have now is actually to get the molecule inside the tube. This is what we are going to address in this picture. So the Poisson total error bar contains on two error bars, or two parts of the error bars is actually one which is partitioning error. The partitioning error is actually limited by the number of partitions, or the number of partitions that you actually can use. So as I mentioned earlier in this presentation, I talked about this, that there are vendors that has different number of partitions. So we have two different. One is 8,500 and one is 26,000 partitions. So they have different partitioning error, because if you can load less in the 8,500, because that will be oversaturated much faster than the 26K. And that is what this partitioning error actually stands for. So as the normal concentration actually increased, that means the number of amplified targets per partition is increasing. And eventually you will oversaturate the well. Then you see that this partitioning error actually goes up. So if you look at this is just two examples where we have 20,000 partitions and 1 million partitions. So the 1 million partitions to the right, you can see that the partitioning area is very, very, very low. So it's very low because we have a lot of partitions, so we can load a lot. We can actually have a very abundant target without saturating the system. But eventually, if we would just load, we will eventually saturate even the 1 million partition. So the partitioning error is limited by the number of partitions that we can actually use. And to contradict that, we have another thing called subsampling error. And this is what I addressed in my earlier slide when I mentioned that you need to get the molecule inside the tube for us to be able to find it and to amplify it. It's one thing that we can detect it, but we also need it to get into this tube. And this subsampling error is what it's all about. So subsampling error actually sets the limit of detection. That means that if, for instance, you're taking a blood sample from me, 5 milliliters of blood in my volume, which I have, which is 7 liters of blood. And of course, if you have a rare mutation that you're looking for inside my body, the chance of you actually missing it when you just take 5 milliliters of blood out of 7 liters is quite big. So that is what's called subsampling. So to really be able to be sure to get that molecule from me into the tube, you need to take multiple samples from me to be able to actually get, let's say, 1 in 10,000 or 1 in a million, for instance, into the tube. So that is actually what subsampling is all about. So as you can see here, the more DNA we put in, the more copies per partitions we have, the less subsampling error we have. So that means the more DNA we screen, the lower limit of detection, the lesser subsampling error we get. And as you can see on the right, when we have 1 million, and we just add DNA, the subsampling error basically goes to 0. So that means that the partitioning error and the subsampling error is somewhat contradictory to each other. But this is statistics. But this is for all digital PCR platforms out there using Poisson statistics. These are rules that you cannot ignore. If we go to applications, tips and tricks, and hallmarks of PCR. So what is the applications normally used for digital PCR? Yeah, of course, mutation detection is something that you immediately think about because, of course, this is one of the major things. But I would perhaps say that mutation detection, yes, but more like rare event detection, because when you have something rare that you are looking for, and that can be anything basically. It can be a virus. You can look for invasive species like environmental DNA or ancient DNA, something that you just can't find with a normal standard qPCR or a normal standard endpoint PCR today. Then, of course, as you have seen with this partitioning possibilities and the amount of load, a lot of DNA, and you can go this low limit of detection. Of course, mutation detection is one of the things that are like highest on the chart. Copy number variation is also one of them. And this is because you can see very small differences you have precision of plus minus 10%. I will come back to that. But that means you can actually see differences in higher copy number. And this is something you cannot do in a normal qPCR. So what that means is that you can see a difference of copy number of 10 and copy number of 11. And this is something you cannot do with the qPCR. When we talk about gene expression, this is actually quite not so straightforward as many things when it comes to digital PCR. Because normally in gene expression, you have cDNA, and cDNA you have very abundant, you have quite a lot of cDNA. That means that normally when you use the references that you have on a qPCR, they tend to saturate the whole system. Again, the partitioning error comes into play. And that means if you, and I will come back to that as well, you need to perhaps switch your reference genes. There are also some other application on this list, but as I said, if you just use your imagination, anything you can extract DNA from, anything you can create an assay for, you can do digital PCR on. If we look at lower limit of detection, and this is again about the subsampling. So this is for human, so this can be different in different ways. As I said, the only thing in digital PCR that's different from a qPCR is that you normally don't do replicates. But there are some occasions where you actually need to do that. And that is when you need to lower the subsampling error, when you screen for something really, really rare. And this means that you need to screen so much DNA that you actually cannot fit that into one well. That means you need to split your sample up. So as long as for a normal mutation, for instance, with 0.1%, when you want to find 1 in 1,000, of course, 1,000 grams of DNA is sufficient enough and you can use one well. But there are occasions where you, let's say, want to screen 1 in 1,000. Or 1 in 100,000. And of course, you need to screen a lot more DNA, let's say up to 1 microgram of DNA. And then, of course, you need to actually do a split of the sample because it's just too much to fit into one well. So then you need to split it into 4 wells instead in this case. So this is where you start to do, let's say, replicates. But of course, you can see these points as one well in our software. We call that a hyperwell. So you can actually just combine them into replicates, and the software will calculate that as one data point. If we talk about next generation sequencing and digital PCR, so this is a very good match, a good marriage, if you will. Because normally, next generation sequencing, if you use MiSeq, HiSeq, or Ion Torrent, or NovaSeq, normally you can go down to, let's say, 1% or 0.1% at best. It depends on how deep you do your sequencing. But of course, when you are down to, let's say, 1%, 0.1%, a normal qPCR is not good enough to validate what you get. If you are within 1 to 5%, you can use a real-time PCR. That's not a problem because it can detect down to that, have that precision detected down to 1%. But of course, when you have the QIAcuity systems or digital PCR, you can go down to, as I said in the previous slide, to 0.1, 0.001, and even further. So you're not limited to how low you can go. As you just heard, how low you can go depends on how much DNA you have. But digital PCR is also more cost efficient than next generation sequencing. Sequencing is good when you don't know how the sequence looks like. Then it's very nicely for you to actually do the sequencing. You get the sequence, then you can create an assay, and then you use that assay to screen with digital PCR, which can actually detect it to very low allele frequency, or if it's a rare sequence, or if it's gene expression, for instance. If we look about how you can normalize a sample input, so this is actually a way of doing that. If you, let's say in this example, this is human DNA, and this is mostly for people that are working with, let's say, plasma. You don't want to waste your sample because you have so little samples, you don't want to waste it. So in this case, what you can do is actually just use what you have totally amplified inside your well. So let's say we have a well like this, that we have 100 copies of target A, and then you have 1,000 targets of target B. And of course, the total amplified target molecules is actually 1,100. The total, so then you take this times the reaction volume that we have, which is 30 microliters in our case, then you know that you have 33 copies in total. And of course, if you use that number times the haploid genome, which is 3.3 picograms of DNA, then you are up to 108,900 picograms, which is 108.9 nanograms. So the theoretical lower limit of detection in this case, using the rule of 3, screen 3 times many molecules, for the lower limit of detection, then this means 11,000, which means in our case, if we have this many, this sample input, we have 1 in 11,000. So, and this means assuming no false positives. And of course, the false positive rate is something you need to consider if you look for mutations. And what you need to do then is that you need to screen your assay with wild types. So you need to screen at least, I would say, a plate with the wild types to see if you have an assay-specific false positive rate. And this means that these false positives should come up randomly in the wild type. If they are just coming in one sample, then this is not a wild type anymore. So if we look at copy number variation, so QIAGEN do this in a very simple way, because we are using a reference genome in one well, and we are using that as an equivalent. So we know that our reference has, let's say, 20 copies per genome or 2 copies per genome. And then we use that as an equivalent to calculate the copy number variation. So if you take example 1, where you have your reference sample has 2 copies per genome, and target A has 1,000 copies per microliter as an absolute quantification, and target B has 5,000 copies per microliter of our reference locus. So this corresponds to a ratio of 0.2, and is equivalent to 2 copies per genome, because this is the reference that we have. So this is what we use. So target A, in our second example here, target A has 1,500 copies per microliter, and target B has 5,000 copies of your reference locus, just like the other one. this corresponds to a ratio of 0.3 compared to your known reference sample, which we know has 2 copies per genome, and 0.2. We just divide them, and then we see that 0.3 divided by 0.2 times 2 copies is actually 3 copies. So this is a way of using just one reference for, let's say, a whole plate. So this is a little bit different. Of course, you can also do it using the absolute quantification when you have CNV equals the n times the concentration of the target divided the concentration of the reference, using a probe-based approach as well. If we look at gene expression, and this is where I was referring to a little bit before, that if you use your old references and there has been some issues coming forward when you do this, because you tend to oversaturate the system with your old reference genes. So when you start a new project with gene expression, so my suggestion to you is that you run your 2 conditions on a qPCR first with only your reference gene, because then you can pick the one that are in the Goldilocks zone for the reference gene, if you will, but somewhere I would say between 25 to 30. If you can find a reference gene between CQ or CT of 25 to 30, which is stable in the 2 conditions, that's the one you can use in digital PCR. Because anything below 21 here on this side down to 0, it's too abundant and will saturate the whole system for you when it comes to gene expression. However, when we talk about the resolution you will have gene expression is outstanding with digital PCR, because we talk about plus minus 10% unparalleled precision. What does that mean? Well, if you see the blue area here in the presentation, that equals the difference you have in the CQ value with a 2-fold dilution in a real-time PCR. And as you can see, if you do a 10% dilution of your sample with a spike in, for instance, like this, you see that you have many more data points. You have 1, 2, 3, 4, 5, 6, 7. So up to 7 data points more there where you actually can distinguish and see a difference in gene expression. And this is particularly interesting when we talk about microRNA, because you can see microRNAs for biomarker screening has started to grow, and people tend to want to see if there are difference in microRNA expression, especially in serum and plasma. And of course, sometimes the difference is too small for a qPCR to detect. And by looking at it with digital PCR, you are able to actually see these small differences in a much higher resolution, and they can have a significant impact on the biological response. Of course, when it comes to normal gene expression in qPCR, this small, let's say, differences may not be biologically relevant, but hey, this is up to you to decide. But as you can see, 2-fold dilution inside a digital PCR compared to a real-time PCR is quite high resolution, because either you have 100 copies per microliter or you have about 50 copies per microliter. So you see it's very high. As you can see, you can for sure get a lot of more data points inside, and this is quite nice to see. So when do you want to do digital PCR? Well, that is entirely up to you, I would say. No, but to be honest, as I mentioned before, if you have recently or previously ran into issues, stopped a project because of too crude samples, due to extraction problems where you get very low yield, or let's say you cannot perform a PCR in a good way, or a real-time PCR because the efficiency has been just too bad and the data has been looking, well, not good enough to present. Well, Now it's the time to take those old projects and blow the dust away and take a look at them again. Because with digital PCR, with the resolution you will have, the precision you will have, and since it's an endpoint measurement, these tough samples that you have problems with before you will be able now to actually perform the PCR and perhaps generate really, really good data on this one. So, but it's all, what you need to think about is that you need to be able to do an assay. And if you don't have that assay, if you don't have the sequence, as I mentioned before, then you need to actually sequence the DNA to be able to get the sequence you can create an assay. But of course, it's also important that you have a good extraction method. And QIAGEN has a variety of extraction kits for different and tough samples, but this is crucial. So of course you need to first be able to extract DNA or RNA, so you can actually either do cDNA or make a library for sequencing. And of course you need to do and be able to perform an assay, so you can actually create an assay and perform the digital PCR. But as I said, anything you can extract DNA from and create an assay, you can do digital PCR. It doesn't matter if you have crude samples anymore because of this partitioning, as I have been talking to you about today. When it comes to primer probe design, so what kind of path should I follow? And this is really important. You should always try to follow the hallmarks of PCR, regardless of that this is an endpoint PCR. So that means that you should try to keep the amplicons as short as possible, anything between 60 to, let's say, 250 bases, because that's where the efficiency, because you still need efficiency, because you need an amplification. If the efficiency is 0, you have no amplification, and of course, we want the best possible efficiency. The primary probes should be as in the qPCR, so they are 18 to, let's say, 25 bases in length. And the TM for the probe should be between 5 to 10 degrees higher than the primers. So, because if the primer binds in before the probe, you have dark PCR, and that is not what we want. And of course, the GC content should be between 55 to 65% for optimal annealing temperature. And 3 Gs or Cs in a row should be avoided due to van der Waals bondings. So so we can actually have a good efficiency and good annealing. And of course, if you have very fresh DNA, or if you have plasmids or something that are super coiled, then of course, perhaps for digital PCR, it can be good if you cut the DNA into smaller pieces just to increase the efficiency and make everything more efficient. The most important thing is to use a restriction enzyme that doesn't cut within the amplicon. So now we have come to the end of this seminar, and I am done. I thank you so much for your time. I hope that you will have good questions. I hope that you can put them in, and then we will answer them as soon as possible. But it has been a pleasure for me. And I need to add, in these Corona times, that I give you all praise to your family and friends, that they are healthy and that you all are safe. Thank you so much for me.